What's going on, farms? What's going on, everyone? Hey, Danny, can you hear me? If you guys can hear me, please let me know. If you can hear me, let me know. What's going on, guys? Oh, cool. What's going on, everyone? So we have about 6 minutes to the event. You guys can easily see the event countdown. We have about 6 minutes. So the market is open right now, but I have to be here with you guys. I can't really, trade right now. Please share it with everybody. Let everyone know we are starting very soon. So what's going on, everyone? It's your boy Dayo. This is Frunkla Investment. Today I'm going to be your host. Mr. Invest a lot is not going to be available today, so he gave me the job to host you guys today on this event of host genome analysis of structural aberrations by optical genome mapping in patients with severe C19. So that's why I am here right now. What's going on, everyone? It's going to start in the next 4 minutes. I am live. I'm trying to invite as many people as possible to come and join us. This is my third time of going live on YouTube, guys. I mean, it's not easy. I don't understand how Mr. Invest does it. I don't know how that guy do it almost every time. It is not easy. What am I have to 6 5 3 hours to go on YouTube. I want to share it with okay. So what are you guys doing today? Are you buying more shares of Bionano Genomics? Are you holding, taking profit? Let me know what you're doing. Let me know what you are doing. It's going live in the next 2 minutes. We have like 2 minutes 45 seconds left. So I am I am excited to hear what Dr. Ravindra is going to say about, what he has been able to do with the Sapphire device. As you guys can see in the description, he mentioned, of course, C19, and we all know that the pandemic right now is, destroying a lot of businesses. So hopefully they have some use cases for Sapphire, and they have been able to, or they will be able to use Sapphire, to help detect what is going on with this this illness, this virus. So, oh, what was that? So we have 1 minute left. We have a minute left. We have a minute left, guys. I have to reset this a little bit. I have to check the price of BNGO right now and see what is going on with the stock price, because I have to manage my internet so that this connection is not, disturbed. So what's going on, everyone? So we have less than a minute left to the event. What's going on, everyone? It's your boy Dayo hosting you today. I am super excited about Bionano Genomics. You guys know this is my number one company, my number one investment, all in BNGO. I am super excited about what the device is able to do. We have to find solution. We have to help researchers find, cures for these rare genetic diseases, cancer, and so on and so forth. So we are going live, guys. Watch webinar. Okay. You guys are funny. Mm. I am doing good, guys. I am wonderful today. And how are you? Thanks for asking. I think they are going to start in the next 4 minutes. So four o'clock. So I think we are starting. The music has stopped.
Hello everyone, and welcome to today's webinar, Host Genome Analysis of Structural Variations by Optical Genome Mapping in Patients with Severe COVID-19. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Bionano Genomics. To learn more, visit bionanogenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Ravindra Kolhe, Vice Chair of Pathology and Section Chief of Molecular and Genetic Pathology, and Director of Georgia Esoteric and Molecular Lab, Augusta University, and Nikhil Sahajpal, Research Associate, Georgia Esoteric and Molecular Lab, Augusta University. Dr. Kolhe, you may now begin your presentation.
Thank you. Good morning, everyone. My name is Ravi Kolhe. I'm a pathologist at the Medical College of Georgia, Augusta, Georgia, the place of masters. Welcome to the seminar. What we're going to try to do today is present or attempt to understand the structural variants in individuals who are severely affected by COVID, and the importance and use of very appropriate technology for something like that, which is optical genome mapping. So these are my relevant disclosures for the contents of today's talk. Most importantly, I'm employed by the state of Georgia, and the opinions which I'm presenting here are personal and does not reflect the opinions of my collaborators or my employers. And most importantly, this is a Bionano sponsored webinar. So if any use of Bionano product described in this presentation may not have been cleared or approved by FDA for any applicable or any other applicable regulatory body. So some introduction to the lab. So I was recruited at MCG 9 to 10 years ago to start the molecular program. The lab which we have put together is a CLIA lab. Mostly we do complex esoteric testing, which has really helped us to bring really cool and cutting-edge technologies in clinical space. Before I start on today's data, I want to take a step back and put together what we have done for the last 1 year for the COVID, and not just us as individuals, but as a collectively lab medicine folks. So many of you know that till end of February last year, CDC was the only manufacturer and tester of CDC was the only manufacturer and tester of the COVID-19 in USA. And then after that, give me 1 second, I think there's some technical issue. Okay, can you see the slides moving? I think there's an issue with the slide control. Okay. Okay, you can go ahead and move the slides. I think you can take control and move the slides, and I have my visual thing. Thank you. So just to give some background, so I'm on the slide 5, the FDA UA validation. And what we ended up doing was we validated multiple different aspects of COVID testing to help and make sure that what we were trying to do was reaching many people. The validation process or the EUA validation process was not easy. We and my lab, especially Nikhil, Alan, and a couple of people in the group, and Ashish, we sat together and basically dissected the EUA application and then put together a program which really helped us submit all the information to FDA. And it was, I think, a year ago, March 13th, we were able to submit all the documentation to the EUA. And on March 15th, 2020, we got a letter from the FDA to go ahead and start working with this. And we went live at that particular night, and that has really helped the region, the state, and then probably the Southeast United States, because we were one of the few labs to get things started on that. And after that, for the last 1 year, the lab has pretty much transformed everything we're doing into something which was mostly COVID focused. Each and every aspect of the COVID testing, we have worked together and made substantial changes working with our collaborators as well as FDA to make all those changes happen. The biggest impact I think we were able to put together was the help in supply chain constraints and its optimization. And we pretty much worked changing from the sample collection from the swab to the way the samples were collected in the VTM. And most of all this is published now in multiple articles with Nikhil's efforts and then the entire lab's effort. One of the, I think, the most impactful thing we were able to put together was use of saliva as a sample type. And I think we not only put together as a sample type, but we were able to use and show and validate that saliva can be used not only as a sample, but for pooling as well as without RNA extraction, as well as a multiplex for looking at influenza A and B. Okay, with that introduction to our COVID work, I want to dive into slide 15, which basically talks about the pandemic and what we have done to help us understand the role of structural variants in severely affected COVID individuals. So as many of you know, this probably is and will be one of the biggest challenges, at least in my lifetime and our lifetimes. More than 150 million cases, 3 million deaths. And we're still struggling to understand what exactly caused this differential split survival, not only in the symptoms, but also the ICU or the oxygen requirement, as well as the mortality in these individuals. So this was around May of 2010, when I was talking with some of my collaborators, including Alka Chobe, and we were looking at in our clinics and ICUs, we would have these hundreds and thousands of patients or individuals who were infected, who were basically absolutely asymptomatic. And on the other hand, we had a small percentage of individuals who were infected, but who were not only infected, but severely symptomatic, and some even required oxygen support, and very few of them end up dying. So the idea was to understand if there is something in the host genome which is making all this susceptibility to the severity of the symptoms, as well as the mortality and outcome of the infection. Because at that point, we knew that it was not the viral load, as usually in cases of infection, especially viral-based infection, the viral load sometimes can dictate symptoms and outcomes. But in SARS-CoV-2, we know that individuals who are having low CT values or high viral load were absolutely asymptomatic. So one of our hypotheses was there's something in the host genome which is making this huge split survival and impact on mortality. And this discussion led us to forming a very organic COVID-19 host genome consortium, which is highly focused on looking at structural variants. Because when we're discussing this, we realized that there are multiple aspects of these individuals who were coming into ICU, have comorbidities, especially focused on diabetes, hypertension, heart failure, but many of these deaths were extremely unexplained reasons, with many explained reasons. There are multiple other COVID-19 host genetic consortium. We're looking at mainly the whole genome. And we reviewed the data and looked at their findings, and we identified some of the challenges or limitations of those consortium and the data which they have. One of the biggest limitation was they were not able to detect the larger variants. And that basically helped us to put together the COVID-19 structural genome together. It's a global consortium with more than 30 plus labs, with leadership from me and Alka, and we're trying to get as many people as possible involved with the whole variety of expertise which are helping us dissect this unique hypothesis based on split survival in COVID-19. And as you can see, the labs are all across the consortium. There's a global reach, and many of the labs are not only US but outside US which are participating. The principal investigators come from all across the globe with a variety of expertise in understanding human genome. And I'm very grateful that we were able to put together this amazing group of people who are trying to help us dissect this particular problem. The objectives of this particular consortium is fourfold. One of the main things we really wanted to do was create a global network of labs who are using novel long-read molecule technologies to assess the structural variants in host genome, and then which can help us understand these questions. And again, this hypothesis or this interest or question arises from one of the major challenges whole genome sequencing was not able to provide based on the GWAS studies on the host genome or host genetic studies. The idea also was to create a global network of labs to share the data, to share the samples, as well as share the results and approach in understanding this particular aspect of COVID-19 host genome. The other objective we have in the consortium was to create a global forum to share all the data we have generated from different genomic methods, not just one, but multiple long-read molecules, and compare the data, help create different bioinformatic tools, and then different methods, and hopefully make it readily available for everyone. And when we started looking at the initial screening data, one of the things which are really helpful to understand was the role of the structural variants in contributing to the host genome, mostly focused on two aspects. One was the immune response, as well as the viral replication factors. So Nikhil will probably go in detail on the data aspect from the genome as well as the RNA side to make that case. But the consortium has helped us to shed light on genomic research to search for more informative structural variants for common polymorphism, protective genome variation, and other factors, especially which can other genomic factors that contribute to mostly the SARS-CoV infection, progression, and recovery. And as I said, this is a very organic, open platform and group. All this data will be publicly available based on individual institutes, IRB, and research initiatives. And one of the other things which we are really trying to put together is using optical genome mapping. And I think it's very underutilized in something which is for something like understanding immune response in COVID-19. And this consortium has really helped us to put that aspect together. This effort is not something which I can do on my own. There's a whole group of people in my lab which are working day and night, not only for the host genome effort, but also for the entire SARS-CoV-2 response, which we have in the lab for the region and the state. This is during some part of pandemic. The different members of my lab who are working. One of the main people behind this is Dr. Mondal, which is the lab supervisor, but not also very well supported by Dr. Chaudhary and DuPont, which help me make sure that I'm not doing something really out of the box, especially when it comes to clinical care. Three very important ladies in the lab, Kimia, Sudha, and Yasmin, which are from three different groups of families, but are working day and night to help us keep the SARS-CoV-2 response up and going. But not the least, one of the smartest people in the lab, Alan, Nikhil, Pankaj, and Max, which are helping put together the scientific aspect. So with that, I would like to invite Nikhil, who is the driving force behind most of the data which you will see for the COVID-19 host genome. We were able to put together a manuscript which has focused on our initial screening data. It's in preprint, and Nikhil will go over the data, and then I'll join you guys back for the Q&A. Nikhil, you want to take over?
Yes, thank you so much. I will be happy to present our study which incorporates the analysis of structural variations by optical genome mapping in patients with severe COVID-19. As Dr. Kolhe already mentioned that there are several host genome investigation consortiums and efforts going worldwide, but all those efforts are majorly focused on using the sequencing variants for analyzing the host genome. Thus, the structure variations or large structure variations are rather undocumented for the insights in COVID-19 predisposition. In general, structure variants, including copy number variations, account for a significant proportion of variations among individuals. Thus, the aim of the consortium and the study was to investigate the host genome of individuals with severe COVID-19 at level of large SPs, which is 500 base pair and above, in order to identify events that might provide insights into the inter-individual clinical variability that we are seeing in these patients infected with SARS-CoV-2. We have so far mapped around 100 genomes, but I will be presenting results from the first 37 severely ill patients. These patients were recruited in the first phase of the pandemic, and the inclusion criteria were the patients that were positive for SARS-CoV RT-PCR test and required mechanical ventilation or a fraction of inspired oxygen. The mean age of the severely ill group was 60 and was dominated by African American ethnicity. These patients had chronic pre-existing medical conditions, and the genomes were mapped using Bionano Sapphire system with the HG19 genome assembly. The Sapphire workflow relies primarily upon isolation of high molecular weight DNA, and in this case, the DNA was isolated from the blood samples from these patients. Once the DNA is isolated, the DNA is labeled at specific 6 base pair motifs. The labeled DNA is then loaded onto the chip, which goes into the Sapphire instrument. Inside the instrument, the DNA molecule linearizes based on electrophoresis as it passes through the microchannels and nanochannels. And while it is passing through the nanochannels, the images are captured. These images are used to formulate a de novo consensus map, which can be aligned to any reference genome, in this case HG19. And the difference in the labels is used to call out structural variations, and there is an additional copy number track which is based on coverage. For the analysis of data once the genomes are mapped, we use 3 different approaches. The first approach focused on analyzing or identifying real unique structural variations, for which we did an unbiased whole genome investigation that did not appear in any of the control database. The Bionano Genomics control database is around 300 samples strong, and the SVs were looked that were not present in any of those structural control samples. Further, the structural variations were filtered for those SVs that disrupt the coding region of the gene. Further, only those structural variations were interrogated which were implicated in immune response or infections in the literature. The second approach focused on identifying common structural variations. For this, a manually curated gene list was identified or formulated, which included around 850 genes, which were genes that were implicated in SARS-CoV-2 or viral infection or immune pathways. Then the structure variations overlapping these genes of interest were identified, which also appear in less than 20% of the Bionano control population. Further, only those structural variations were evaluated which disrupt the coding region of the gene. The third approach utilized genome-wide HV enrichment in severely ill population versus the control population. In this, the data was enriched by filtering the number of cases for each gene more than 2 and filtering for HVs that appear in less than 20% of the control population. The top ranked genes were identified using odds ratio. I will go into each of these analyses in the following slides. The key aspect of this analysis is actually the rare unique SVs, and we identified 7 unique rare structural variations impacting 31 genes. The most important of these is rather STK26. Here in this slide, we can see a copy number track where a gain in copy number is observed, whereas in the structural variation track, we can see the copy number gain is a tandem duplication. If we see closely at STK26, we see that the duplication duplicates partially or overlaps partially the STK26 gene, which is actually the 5-UTR and coding exon 1 of this gene. We confirm the structural variation gain by qPCR experiment with a simple dosage experiment where we use 4 controls, 2 males and 2 females. And you can see the patient with the duplication in blue, patient 44, and 2 other patients which did not had the structural variation in black. The gain in function is shown with a dosage gain above 1.3. Interestingly, STK26 is a negative regulator of the TLR pathway. TLR pathway has been implicated in severe COVID-19 cases with both whole genome analysis and RNA-seq experiments, where loss in function of TLR7 and TLR3 pathway has been implicated in patients with severe illness, and RNA-seq experiments have shown that type 1 interferon response is reduced in patients with severe illness. So STK26 post-translationally inhibits TRAF6 and inhibits the TLR pathway. To investigate the functional relevance of STK26 gene, we looked at the literature and found that since the duplication was in the 5-UTR region and coding exon 1, in the literature we found that dual 5-UTRs are actually synthesized in synthetic biology that result in increased transcript and translation of the gene of interest. Consistent with the hypothesis, we found that in expression studies, STK26 was upregulated in all the severely ill patients compared to asymptomatics, whereas the patient having the duplication was the one with the highest expression. The next step, we wanted to look at the expression of especially the STK26 post-translational partner, which is TRAF6, and we found that even TRAF6 is significantly upregulated in severely ill patients compared to asymptomatics. Even downstream IRF3 was significantly upregulated in severely ill compared to asymptomatics. However, since this interaction is at the level of protein, if STK26 upregulation is inhibiting this pathway, we should see expression level changes at the level of type 1 interferons. Consistent with the hypothesis, we found that ISG15 expressions were significantly lower in severely ill patients compared to asymptomatics, while the patient with the structural variation was the one amongst the lowest. Similarly, if one expression was significantly lower in severely ill patients compared to asymptomatics, again the patient with the structural variation was one amongst the lowest. So this data actually implies that the duplication of the 5-UTR region led to an upregulation of the gene expression, and this interrogation led us to this coincidental finding where we found that STK26 is upregulated in all severely ill patients, but because of the structural variation, the effect was potentiated in that patient and could have caused the severity in that patient. Additional evidence in the literature comes from one of the senior consortium members, Dr. Alexander Hoyshin, who had shown previously that a loss of function of TLR7 variant led to reduced type 1 interferon response and was associated with severe COVID-19. Most recently, a JIVA study from an Italian consortium showed that a loss in function of STK26 variant is associated with protection in males, which is again supportive to our hypothesis, where we have found gain in function with severity and they had found loss in function with protection. Coming to the second structural variation, this is a heterozygous deletion of chromosome 2 that partially deletes DPP4 gene. If we see more closely, we can see that these three labels are deleted in the Bionano map and they actually overlap 3 exons. So the deletion over the 3 exons was confirmed by qPCR dosage experiment, where you can see that the patient 38, which is the one with harbors this SV, the dosage drops to 0.5 for both exons 3, 4 and 5. Now DPP4 is a membrane-bound receptor and is actually the receptor for MERS entry into the cell. However, the cell line experiments have shown us that it does not serve as a receptor for viral entry for SARS-CoV-2. So what additional function does this receptor have? Many of the cleavage enzymes such as matrix metalloproteinases cleave this enzyme, and the DPP4 exists as a soluble form in the circulation as SDPP4 and has several immune effects such as T cell activation. So we measured using ELISA the soluble DPP4 levels in the serum of severely ill patients and compared with asymptomatics, and found that the soluble DPP4 levels are downregulated or reduced in the severely ill patients. But especially if you can see the case 38, which is the patient with the structural variation, the level is significantly lower as compared to even other severely ill patients. The additional support to our finding comes from another published report, where this group had also shown that the soluble DP4 levels were reduced in severely COVID-19 patients, but they also showed that the effect was specific for SARS-CoV-2, that the soluble DP4 levels do not reduce in patients with sepsis or other infections. The third SP is a heterozygous deletion of chromosome 16 that contains 6 genes, most important of which is the gene VDHHC1. Again, the structural variation was confirmed using qPCR dosage experiment, and you can see the dosage dropped to 0.5 for the patient with the structural variation. VDHHC1 is again implicated in type 1 interferon response, where it stabilizes by a post-translational modification of IFID-M3. IFID-M3 guides the endosomes and stabilizes the endosomes to lysosomes so that it impedes the viral replication. A deletion of VDHHC1 can be hypothesized as a loss in function and thus a loss of this hypothesis and this pathway. The next structural variation is a duplication of chromosome 17 that includes 2 genes, KRT15 and KRT19. Again, the structural variation was confirmed with qPCR dosage experiment, where we can see the dosage gain on the patient with the structural variation. The KRT15 and KRT19 are cytokeratins that are bound at the cellular junction, and they enable the viral transmission through cell-to-cell viral transmission. So a gain of the entire gene of KRT15 and KRT19 would rather can be hypothesized as a gain in function and would predispose to higher cell-to-cell viral transmission. The next structural variation is a duplication of chromosome 17 that includes 15 genes, most important of which are the CD300 cluster of genes. Again, the structural variation was confirmed by qPCR dosage experiment. What is interesting is that CD300 are immune receptors on immune cells, and these have been exploited by the virus to evade host defense and help in viral replication. So a duplication of the entire region leads to, can be hypothesized as a gain in function, and thus helping the virus to evade the host defense and leading to viral replication, and thus leading to severity in these patients. The next structural variation is duplication of chromosome 19 that includes the entire GNF443 gene. This is actually a very interesting gene. This is one of the genes which has a sequence homology to SARS-CoV-2, and the homologous sequence genes have been shown to help the virus to evade the host genome, immune response, and help in viral replication. Again, the duplication was confirmed by qPCR dosage experiment, where we can see the gain in dosage of the patient 13, which has the duplication, where the dosage goes to a dosage of 2. The next structural variation is a duplication of chromosome 1, partially disrupting 2 genes, which include ADORAT gene. Again, although a duplication of an entire gene can be correlated with a gain in function, but here we see that the duplication is disrupting this gene, where the duplication event starts from intron 4 and duplicates exon 5 and 6. Again, the duplication was confirmed by qPCR experiment, and this unique event was identified in 2 patients. So ADORAT gene is a promoter of NF-kappa B, which is an important immune player that helps in has several responses, which includes antiviral response. So these were the 7 unique rare structural variations that we identified in this investigation so far. The next aspect of the analysis was identifying common structural variations. These include 4 structural variations that impact 6 genes. The first structural variation is an inversion of chromosome 3 that disrupts the MUC4 gene. Interestingly, mucin genes are the genes that produce mucus, and they are the first line of defense against an invading pathogen. So the mucus entraps the virus, and through the mucociliary function expels the virus, and the T3 class of mucins, which includes MUC4, elicit the first immune response against those viruses in the epithelium. So the disruption of MUC4 can be hypothesized to lead to loss in function, and in literature there is extensive documentation that loss of MUC4 or null MUC4 mice exposed to different viruses, including SARS-CoV-2, lead to severe lung infection, pneumonia, and death. So this MUC4 structural variation inversion was overrepresented in our cohort, which supports that this structural variation is implicated in severe disease. The second common structural variation is a duplication of chromosome 17, which includes the CCL3L1 and CCL4L2 gene. Interestingly, this duplication is in the CNBR region, the copy number variable region, and the coordinates vary for different individuals, but they always almost include both these genes. The structural variation in all these patients were confirmed by qPCR, and you can see the different dosage as it is a copy number variable region. The CCL genes are actually immune genes and inflammatory genes. So in literature, in severity in COVID-19 patients, we have found that the CCL3L1 and CCL4L2 genes are upregulated both in the epithelia and in the BLF fluid. So a duplication of this region and the overrepresentation of this structural variation in our cohort compared to the controls implicate this structural variation in severe COVID-19. The third common structural variation is a deletion of ApoBec genes, and these are the viral editing genes. So a loss of a viral editing gene can be seen as a formulation for the virus to evade the viral editing or the immune response. Again, the loss was confirmed in these patients with the help of qPCR experiment. The fourth common structural variation is an insertion in the promoter region of MX1 gene, which is a type 1 interferon. Evolutionarily, what we have seen that as the length of the 5-UTR or the promoter region elongates, it leads to reduced transcription of the gene. So in RNA-seq experiments and single-cell RNA-seq experiments in the literature, almost all these literature predominantly finds that the MX1 is downregulated in severely in patients as compared to population of controls. So an insertion in MX1 gene and the overrepresentation of this structural variation in our cohort compared to the controls is worth of an SP to follow up with expression analysis and seems to be implicated in patients with severe COVID-19. So I would like to conclude the findings that the patient-centric investigation identified 11 SPs involved in 38 genes implicated in 3 key host viral interaction pathways. The first is innate immunity and inflammatory response. The second is airway resistance to pathogens, which includes MUC4 gene. And the third is viral replication spread in RNA editing, which includes the ApoBec enzymes. Most importantly, we find that STK26 and DPP4 are the most promising candidates, both based on the rarity of the structural variation and the expression analysis and the support from the literature and with the genetic analysis that is coming on from other GWAS experiments. These findings actually extend the previous reports that involve innate immunity and type 1 interferon response in the pathogenesis of COVID-19. And most importantly, we find that optical genome mapping is a powerful tool to identify large structural variations, which must be documented to investigate the host genome to identify the predisposition of or susceptibility to severe COVID-19, as we are looking at the vast inter-individual variability associated with infection in patients infected with SARS-CoV-2. To summarize, we had put up this schematic to represent different pathways and put out a holistic picture to what we are seeing, and we actually have incorporated the major structure variations, and you can see the STK26, DPP4, ZDHSC1, and a diminished response to type 1 interferons. And most of these players actually overlap with different pathways, where, as you can see, that ZDHSC1 translationally modifies IFIT MC, but also downregulates the type 1 interferon response, and so on and so forth. So with this, I would like to acknowledge especially Dr. Ravindra Kolhe for leading this study and the consortium, Dr. Ashish Mandal, who is our laboratory supervisor, Dr. Alka Chaube and Dr. Alex Stacey at Bionano Genomics, Dr. Rashmi Kanagal Shamna at NG Anderson Institute, Dr. Mike Zodi from New York Genome Center, Dr. Eric Jarvis from Rockefeller University, Dr. Ellen Beggs and Dr. Kathleen Bounchy at Harvard University, Dr. Alexander Hush at Cranbourne University, Dr. Vineet Bafna at UCSC, Dr. Bryn Levy at Columbia University, and Dr. Silvio Ellen Pequeno at Virginia Commonwealth University. Thank you so much for your attention.
Well, thank you, Dr. Kolhe and Nikhil Sahaj for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located in the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, let's see. Sorry, they're moving around. People are asking questions. That's great. Okay. Why and how is the hypothesis of host genome variations, SNPs, SVs, is crucial for severity of the disease? Why not the viral genome variation?
Can you hear me? It is Ravi.
Yes, we can hear you.
Okay. So when we looked at the disease biology, I mean, right from the January 2020, there were multiple factors which are playing a crucial role in determining who is becoming symptomatic, who is becoming oxygen dependent, who is becoming ICU dependent, and who is dying. And we looked at all possible aspects of those things. One of the biggest things was not even viral genome. You can talk about the viral load. The biology will dictate if you have high viral load, you should be more symptomatic and need oxygen. But that was not true. When we looked at the original, the Wuhan genome, the B1 or the B, majority of the infections up to May, June, maybe up to July or August were with the original genome. And even with the original genome, we saw such a variation in symptoms, not only symptoms, but ICU admissions, oxygen dependent, and death. So we have a very crucial and substantial evidence to show that it's not the viral genome which dictates majority of these split survival or split symptoms. Yes, I agree. I think viral genome will definitely play a role in the latter half. But if you look at in the earlier part of the pandemic, I think the whole genome played a significant role, and that's what we are trying to understand.
Thank you, Dr. Kolhe. Yes, and our next question, do you think we can infer which patients will be severe in COVID infection with reference to SKT gene duplication in the UTR region?
So I would like to go with that. Certainly the host genome structure variations, especially the unique ones, are the ones which are to watch out for and are the key ones that can predispose to severe disease. And because we have only seen this structure variation in one patient, and 5 UTR region does lead to overexpression of the gene, so we can certainly infer that because the STK26 pathway is already upregulated, a structural variation in that region potentiates further towards disease severity. So this is what we are inferring, that the structural variation that we had seen in STK26 is actually potentiating further towards severity of the disease.
Thank you, Nikhil. Let's see, our next question. Let's see, what is the foremost gene with SVs, which will infer mostly for severity?
So
I didn't hear the question. Go ahead, Nikhil.
Yeah, so the severity, actually you have to see patient by patient. That is why the analysis is patient centric. So any of the rare structural variations impacting or disrupting a key gene in the immune response can lead to severity. So, in this investigation, we have found that STK26, EPP4, or VGHT1, where STK26 and VGHT1 are type 1 interferon response pathway, but PP4 is a different in a different pathway. So they need not be in the same pathway or in the same gene. Like GWAS investigations or sequencing-based investigations have found loss in function of TLR3, TLR7 genes. So it all depends that what it can be a different gene, but if it implicates in a key immune gene or host response gene, then it will lead to severity of the disease. Thank you.
Thank you, Nikhil. Let's see, our next question. Can you see a clinical use for these variants? Can they be used when patients report to the hospital with an infection, or when would they matter?
So, I mean, definitely that is the primary goal of this. I mean, any research or translational research we are trying to do or understand has to translate into clinical use. So the big picture goal or the bigger goal for this study is there a way we can identify structural variants, create an assay, maybe on a PCR or even optical genome mapping, and come up with some kind of early warning, and then predict the outcome of these individuals. So yes, I think these structural variants are definitely critical, and not just in immune response, but if you look at any inherited genetic disease, structural variants do play a critical role in disease initiation, and as well as disease response and mortality. So no doubt about that these structural variants will play a critical role. I think what we are at the current stage we are doing is we are trying to come up with a consensus on which structural variants actually play a significant role enough for us to translate on an assay. And I know there are groups and collaborators in Europe which have already started working on or already in the lab, especially focused on TLR7 based assays. So the answer to this is yes.
Thank you, Dr. Kolhe. We have another question coming in. How can you tease apart whether it is severe COVID pathology causing the structural variant, or it is the existing structural variant causing severe COVID pathology?
I think this has, I mean, if you look at this as a viral infection, I really hope the viral infection or the virus is not causing any structural changes in our genome, because that itself will be a whole different discovery and research. Based on our existing knowledge, our collective knowledge of viral infection, I mean, if you look at influenza or retroviral HIV, there is definitely some damage to the genome by genome integration from these viruses, but I would like to comfortably say that the response to the virus is different based on the structural variant rather than the virus is causing any changes in our structural variants. And I mean, I'm open to discussion and ideas, but as far as we know collectively for the last few years or few decades we're working with viruses, I think it's usually the other way around. Thank you.
Thank you. And our next question, you showed several somewhat common variants, including some only in African Americans. You also showed how African Americans are more likely to be severely affected. Is it possible that some of those common variants you identified may predispose African Americans to this more severe infection?
Exactly.
Yeah.
Yeah.
So, go ahead. I'll love to finish up.
Okay. Exactly. So what we have seen that the common structural variations are actually overrepresented in our data because we are focused on the severely ill, and we have actually written this in our manuscript that because these common structural variations are also overrepresented in the African American population, and we have seen a higher susceptibility of African American population to severe COVID-19. These might be the common structure variations might be a factor that is leading to higher severity in this specific population. So the question already actually answered that yes, it is implicated. It seems to be implicated.
Yeah, the other thing I want to add to Nikhil's discussion is we are in that part of the Georgia where we are heavily dominated by African American population, and when we looked at the number of admissions on 100 Caucasians versus 100 African Americans in our hospitals and ICU, especially focused in South Georgia, we have identified that the mortality as well as the morbidity rate for COVID-related infection was surprisingly higher in African American population. And I think that was one of the reasons we initiated this study, was to identify if there was something which was very specific for the African American population, which is have a higher burden of morbidity and mortality associated with that disease. So again, this data and the study is at very early stage. We are increasing our study population and as well as the control database, and definitely at some point in near future we'll be able to have a solid conclusion on that. But the initial data definitely looks in that direction. Thank you.
Okay, thank you both. So we have a question. Do you see a point in the future where this technology will be applied to examine treatment resistance in clinical trials? Seems very well suited to answer the basic question of was it the treatment or the target that is causing treatment resistance to targeted therapy?
So I would say all of them, because I mean the technology is just a way to identify aberrations. Either it can be related to the disease generation or a target for the disease, or even on therapy response to identify resistance or relapse to the therapy. And I think not only in inherited or constitutional disorders, you're going to see a lot of this technology will show up in identifying the cause of those diseases. But when we come to oncology, it is going to find a lot of new discovery in oncogenesis, especially in comparison to the existing platforms we are using for oncology testing, like karyotype and fission arrays, which definitely have some severe limitations. And in those cases, a platform like optical genome mapping is going to make fundamental difference in our understanding for oncogenesis. And once you identify which is the common denominator for a certain phenotype, then people will jump on identifying therapies for those targets. And in the other group, which the question was asking about the resistance aspect, I think that's definitely when we start looking or dissecting cancers which are on therapy and non-responsive and identify new targets. And in that subgroup also, I think optical genome mapping is going to make pretty severe inputs for us to understand that. Thank you.
All right, thank you, Dr. Kolhe. We have time for one final question. For STK26, the case with the SD has the highest expression. For DPP4, the case with the SD has the lowest expression. But in both cases, you see almost all the severely ill differ from the asymptomatic. Doesn't that argue against the SD being causal if the change in expression is present in anyone who's sick?
So again, we can look at like this or the other way around. So the other way around is that the structure variation led us to the investigation of identifying this pathway, and we found that this pathway is actually implicated in severely ill patients. But as we can see that the patients with the structural variations are the ones with the highest or the lowest levels, which shows that although the pathway is implicated, the structural variation actually potentiates towards severity. So this is where it predisposes to severe disease. Thank you.
Thank you. All right. Well, thank you both. Dr. Kolhe, did you have anything to add, or would you like me to wrap up?
No, I think I agree with Nikhil on that. But definitely, it was a good discussion overall. And I would like to thank your group for helping us put this thing together. We had some initial technical difficulties, but I think it was a great discussion on a very pressing topic on the pandemic. Thank you all.
Thank you. I'm sorry, I thought you had something there to add, to answer that question. So, do either of you, Dr. Kolhe or Nikhil Sahajpal, do you have any final comments for our audience, aside from what you just said?
No, again, I think I can repeat myself. I think you guys have done a great job. This is a very pressing topic, and I'd like to thank the Bionano folks for helping us put this thing together, Alka and Alex and Bill. So thank you. And if there's any additional questions, we will be happy to answer by email. I think both our emails are in the presentation.
Well, thank you again, Dr. Kolhe and Nikhil Sahajpal, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Bionano Genomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
What's going on, everyone? What's going on, guys? So I think that is it for today. Thank you everybody for coming and joining me on the channel. This was supposed to be Mr. Event, but the guy is busy today, probably hustling somewhere, trying to make some money so that he can buy more shares of Bionano Genomics. So I think that is it for today. If you have any question, I can stay for like 2 or 3 minutes and answer any question. If you guys have any question, please drop it. If not, we just have to round up and I can finally go and trade because the market is open, guys. I mean, I have missed like 1.5 hours of action. The first 30 minutes of opening is always the best time to trade. So yeah. Thank you, everybody. I remain your boy Dayo. This is Frunkla Investment. Have a wonderful day, guys. Bye bye.